In this lesson, we'll discuss the idea of experimental design as it relates and compares to observational studies, and we'll also give some terminology that we'll use with experimental design. So with an observational study, you have factors or treatment of interest, but one of the main things about an observational study to remember is that the factors are not manipulated. So you've heard probably many times that you can only establish association, meaning that the two things are just associated with each other. You can't make those causal statements. So for example, if you're sampling from smoking and non-smoking populations and you're going to compare the lung cancer rates of the two groups, you don't have control over who smokes and who doesn't. So again, you don't have the control to be able to manipulate those factors. So if you see a difference in the disease rates for those who have that for those who smoke compared to those who don't, can you conclude that smoking causes lung cancer? And so again, you can't make that conclusion because you cannot control all of the other factors. So maybe there's some other factor that's leading to your inclination to smoke that's also leading to um, the lung cancer rate being higher in smokers versus non-smokers. So again, with observational studies, you're not controlling all of those external factors. So for this type of study, you can only conclude that smoking is associated with lung cancer. So here's a um, common cartoon where, again, you've heard many times that correlation does not imply causation. With design experiments, on the other hand, you are able to manipulate factors or treatments of interest. And so what we can do with these design experiments is when we control those other factors, we can establish that causality. A lot of times, in some cases, you can have practical difficulty in manipulating those treatment assignments. And so sometimes that's why you wouldn't run, say, a designed experiment. So for example, how could we design the experiment that we discussed previously about the causality of smoking with lung cancer to verify that smoking causes lung cancer or lung disease? So what we could do is we could select a group with the same smoking history. So we want to have our initial group as homogeneous as possible. And then we're going to randomly assign each person to be a smoker or a non-smoker. So maybe I'm tossing a coin. If it's head, you're a smoker. If it's tail, you're a non-smoker. Here, if we find that the disease rates are found to be different in the two groups, then we can conclude that smoking causes lung cancer. We've done this randomized trial where we're able to make that conclusion. However, again, the practical difficulty is maybe you don't want to be a smoker. And so if everyone doesn't want to be a smoker, again, you would never get IRB approval for this. It's not an ethical way. Now, there's different studies, case control studies and things like that that they do in medicine. But the point here is that if you can't control those other factors, then again, you can't make that causal statement. Here's another example. So with this shrimp study, they were looking at the best conditions for the growth of commercially raised shrimp. They're going to look at two different factors or treatments. That includes water temperature and water salinity. So for water temperature, we have three different levels, 25 degrees, 30 degrees, and 35 degrees. And for the salinity, we have 10%, 20%, 30%, and 40%. And what we're going to do is we're going to raise these shrimps in containers and weigh them after 6 weeks. So with this example, I want to talk a little bit about the terminology that relates to these conditions. The response here is your observed measurement. Okay, so for this example, that would be the shrimp weights. The treatment factors are the factors of primary interest. And so again, people will call these treatments or the factors, but that's what you're interested in looking at. So in our shrimp example, that's going to be water temperature and water salinity. The treatments are the actual levels. Okay, so again, the treatments are the levels of those factors. And so in our shrimp example, we have 3 of those for water temperature, and then we have 4 different levels of salinity. So again, the treatment factor is the broad factor of interest, whereas the treatments are the actual levels that you're looking at for each factor. It's important to note for experimental design that these levels can be quantitative or qualitative. So quantitative meaning they're 25 degrees, 30 degrees, 35 degrees, or maybe a qualitative factor of cold, warm, and hot. Okay, so you can have different types of factors or treatments. Another concept that we'll cover in this class is the idea of blocking. So again, the purpose of this lecture is just to introduce a lot of the type of designs that we'll talk about in this course. And blocking is one of the factors of your study, and it's identified to account for extra experimental error. Okay, so you're going to try to block out additional sources of variability, and in doing that, the blocks are the actual level of the blocking factor. So in the shrimp example, we don't have any blocking, but we'll talk more about how we introduce blocking later. When you're looking at these different treatment factors, so here again, the different treatment factors, and so we have multiple levels. When you're actually running this experiment, you cross all of the treatments with the for one factor with the other. So that treatment combination here is the cross between each one of those. Okay, so the combined treatment factor is the combination. So again, here we have let's see, we had 3 levels of temperature, right? So let's see, we had 3 levels of temperature and we had 4 levels of salinity. So if we cross both of those, we would have 12 treatment combinations. Okay, so that's how that works with the treatment combinations. Another term that we may use some in this course is the idea of a control treatment, and maybe in your research you've used that as well. The control treatment is a special type of treatment where it's kind of used as a benchmark. So a lot of times in different medical studies, you may be looking at a drug that's randomly assigned. So you have 2 different drugs, but you're going to compare that back to that kind of placebo effect that you've heard of before. So the placebo acts as the control. It's actually it's a drug that's not active, but it's again a way that people can do that to compare back to just that baseline. Okay. So that's just some terminology. Again, we have response, we have some treatments, the overall treatment factor, and then we have also some blocking factors that we'll talk about. In terms of aspects of experimental design, the objective here is to understand the effect of the treatments. So that's what we're trying to do with this design of experiments. We have treatments that we're trying to understand their impact. And in order to do that, we're trying to set up the best design possible to look at that. So we have these different types of designs, and we'll talk about these this semester. A treatment design is just a protocol for defining and applying treatments. Okay, so it's whatever protocol you're doing for that. A subsampling design is a protocol for measuring responses of observational units. So maybe not by the end of this lecture, but in the next couple lectures, and we'll have actually later on in the semester an entire lecture on subsampling, but it allows you to see multiple observations in your study. So that's a different type of experimental design. And then there's error control designs. And for the error control designs, that's where we're going to focus our study. And it's a protocol to identify and account for systematic variability beyond that due to treatment differences. So we're going to talk about how our goal is to figure out the treatment design, where we want to know what's the impact of the treatment, but we're going to utilize these error control designs to be able to efficiently understand the impact of the treatment and take into account the variability that can be associated with other factors in the study. To begin our study, we're going to talk about one-way classification. So what I mean by that is we're going to focus where we just have a single treatment factor. So if you think back to the shrimp example, we had salinity and water temperature. We only would look at one of those. Or maybe we would look at a cross of all of those. So we also could look at this kind of factorial treatment design where we look at the combination of all the levels of those factors. Okay, so again, you could look at all 12 of those levels, but that's more in that factorial structure. There's also a type of designs called fractional factorial treatment designs where you only look at a fraction of those levels of combinations. There's an advantage here that you save resources because you're studying less combinations, but the disadvantage is there's much effort on selecting the combinations, and you have what's called confounding effects. So that means that you can't estimate certain effects uniquely. So if you're interested in the idea of fractional factorial designs or just more in-depth study of experimental designs. We do in the stats department teach a G-level course. It's called STAT 5204G, Experimental Designs. It's usually taught in the fall and spring semesters, and we'll go into much more detail about these factorial and fractional factorial designs. Another design that we'll touch on this semester is a nested design. We won't spend a lot of time here, but again, if you need more help with that, you could definitely see someone in the stats department to help you with this type of design. But the idea of nested is really explaining what's going on. So if you think about it, a nested factor means that maybe with a temperature of 25 degrees, the only salinity that's available at 25 degrees is 10 and 40. So you'll notice that this 40% salinity, there's no way that it can be crossed with the 30. So the 30-degree temperature. For that case, again, one treatment is nested within another treatment. Okay, so levels of this treatment are nested within the levels of this treatment. Okay, and so again, not that you completely understand nested designs after this particular lecture, but that's something that we're going to kind of introduce as we move throughout. Here is a really important topic, and this will kind of wrap up the first lesson here. And it's the idea of experimental units and observational units. And I think that you could argue that it's one of the hardest things that we do during the entire semester. So the first homework that you guys will have will actually cover these two topics. So I want to give you a couple of examples, but I think it's very important when you're starting experimental design to figure out your experimental units and your observational units. So let's look here at the definition. So the experimental unit is the object or objects to which the treatment are applied. Okay, so where are you actually applying the treatments? And the observational units are the objects from which the responses are measured. So make sure you remember that here, it's not the response. A lot of people want this to be the response. So if you think about the shrimp study, it's the growth of the shrimp. That's not the response. You're measuring it maybe from each individual shrimp. So in that case, that's your observational unit, but it's not, again, not the response here. So with the shrimp example, they're going to set the water temperature and salinity for the entire container of shrimp, and they're going to measure the weight of the individual shrimp. So here the EU would be the container, right? Because the entire container here is set to the combination. And then they're going to measure the weight of the individual shrimp. So again, the OU is the individual shrimp. You'll notice here that I'm not saying that the weight of the individual shrimp is the OU. It's actually the individual shrimp. So let's think about that, and let's move on to think about some other examples. So if you're studying human diets, 20 people are assigned a particular diet. After 2 months, we measure the weight loss of each person. What are the EUs and the OUs? So for this one, you could think of with the 20 people, we're going to randomly assign the particular diet that you're going to receive. So again, your EU here, what do we assign our treatment to? Our treatment factor is the different diets. Then our EU is going to be the people, right? And our OU is also going to be the people. And we may be able to get that to come in a lot better than my stylus. But again, that's the, let's see. That's the idea here of the EU versus the OU. So here we're studying shrimp diets. There's 6 tanks of fish diets. There's 6 tanks of fish, and they are assigned a particular diet, and each tank contains 100 fish. After 2 months, we measure the weight loss of each fish. What are the EUs and the OUs? Okay, so let's do this. Let's think about that a little bit further. All right, so we have 6 tanks. 1, 2, 3, 4, 5, 6. Okay, 6 tanks. And in each one, there's 100 fish. Okay, so I'm just going to do a fish, and again, you've got 100, and you've got 100. And the 6 tanks are assigned a particular diet, and each tank contains 100 fish. So let's just say that we have diet, and it didn't tell you this in the study, but let's just say that you have Sketchpad. Just this will work better. Okay, so we've got the diets, right? Okay, and then we're gonna say diet is A and B. Okay, so maybe we've got all of my fish in here, and there's 100 fish, 100, 100, 100, and 100. So I'm randomly going to apply it so this entire container, right, receives each of the diets. So here my EU is going to be the container or the tank, and there's 6 of those. My OU, it says that the weight of each individual fish is measured. So the OU is not going to be the weight loss, it's going to be the 600 fish, right? If instead we said that we were going to measure the weight of the entire container, then that would change so that the OU would be the container. Right? So that's the difference between those two ideas. Okay. So this one's a little bit interesting. If you've ever done split plot designs before, then that's what's going on with this in this example, and we'll touch on those later in the semester. But you're studying 4 irrigation and 2 fertilizing methods. 24 fields are selected for the study. It's much harder to change the irrigation method, so fields are assigned to an irrigation method in a block of 6 fields at a time, while fertilizers are assigned to each field individually. The field yield is measured, and then the question is, what are the EUs? When we said that there are 24 fields, right? And so get the idea, right? Because there's 6. 1, 2, 3, 4, 5, 6. So we just keep going here. And it says that it's much harder to change the irrigation method. So we have irrigation, and let's see, there are 4 irrigation and 2 fertilizing methods. Let's see. So the fields are assigned an irrigation method in a block of 6 at a time, while the fertilizers are assigned to each individual field. So again, with fertilizer, we have 2, and with irrigation, we have 4. So we'll call this A, B, C, D, and we'll call this 1 and 2 for the different fertilizers. So what you're going to do is first you're going to randomly apply. So maybe it's A, B, C, D for these blocks, right? And then within that, you're going to go apply this easy to change factor. So for this one, maybe it's 1, 1, 1, 2, 2, 2. Maybe it's 2, 2, 1, 1, 1, 2, something like that. So the first set of randomizations was to the block of 6 fields. Then within that, we do the second set of randomizations. For this example then, your 1 EU is going to be the block of 6 fields. Right? And then your other EU is going to be the 24 fields, right? So within that, that's the individual fields because that's where we're applying the fertilizer. For the OU, it says that the yield from each field is measured. So for the OU, it will also be the 24 fields. In many cases, your EU and your OU can be the same thing. This one's just a little bit different because you have 2 EUs here. And again, this is called a split plot design, and we'll talk much more about that we'll at least spend another lecture talking about split plot designs. Okay. So let's go back to a little bit more of the kind of terminology. We said before we have this idea of subsampling designs. Okay, so with the subsampling design, it's a protocol for measuring responses of an observational unit. And we think back to the example with the fields. We're measuring each of the yield of each field, and so our observational unit is the field. So often when the case is that our OU is equal to our EU, there is no subsampling. For the shrimp example, if you start with containers with equal weights of shrimp, if you set the water temperature and the salinity of the entire container of shrimp, and the response is the total weight of shrimp in each container, so again, you're measuring the container, not each individual shrimp. In that case, there would be no what's called subsampling because their EU is equal to the OU. Okay, so again, we don't have any of those multiple observations. With the fish tank example, however, the one we talked about with the different tanks, we're studying the 6 tanks and the fish are assigned a particular diet. And we measure the weight loss of each shrimp, or fish, sorry. So in that example, we actually do have subsampling because we have multiple observations, 100 observations per tank. Okay, so in that case, we do have the EU, which is the 6 tanks. We have the OU, which is the 600 fish. So our subsampling here per EU is 100 fish. Okay, so that's a type of subsampling design. The other designs that we're going to talk about are called an error control design. An error control design is, again, a protocol to identify and account for systematic variability. Our idea with these experimental designs is we want to explain things as well as we can. Okay, we want to explain the phenomena that we're looking at. So the idea is everything's in error, right? You start out, you have everything in your experimental error. And we're going to break up that experimental error into 2 different pieces. We have the systematic variability and the unexplained variability. Our goal is to get less unexplained variability because that's the background noise, and that makes for more powerful inference. Okay, so again, if we can explain more of this variability into the systematic component, then it makes for stronger inference and conclusions. So let's talk about one of the types of designs is with blocking. So let's say that you start with containers with equal weights of shrimp. You set the water temperature and the salinity for an entire container of shrimp. And then you have 2 assistants, which each take care of half the containers. But it turns out that assistant A is lazier and tends to give less food to the shrimp than assistant B. Here, the response is the total weight of the shrimp in each container. And we could look at the blocking factor of being the assistant. Okay, so for this one, let's just make it easier. It won't be exactly like the shrimp, but let's say that we have 6 containers of shrimp, and let's say that our treatment, we have the A, B, and C. Okay, those are our treatment levels. Okay, so that might be your cross between your salinity and your water temperature, or we could just call it water temperature. So here, if you pass it out, let's just call this A is going to be hot water. Let's just say hot. B is going to be warm, and C is going to be cold. Okay, so if we pass this out, maybe this one gets an A, this one gets a B, B, A, C, C. So that's just a random passing out of the treatments, and again, your tank here is going to act as your experimental unit. If we're working with that, if you gave this to assistant A and this to assistant B, what's the problem with that? Well, we said that A was lazier than B. So if it turns out that the shrimp, the weight gain for treatment A, right, for that combination of maybe the hot water, if that's actually maybe the weight is actually lower for the hot water, then do you know if it's really because of the treatment factor of the water temperature, or is it just because the the A assistant was lazier and they didn't give as much food to the shrimp. You don't know, right? So a better way of setting this up would be to kind of set up the design like this. So you're going to have randomly apply here, A, C, B, maybe, and randomly apply here. So you did a restriction on the randomization. I'm going to give this to assistant A, this to assistant B. This acts as block 1, this acts as block 2. So what blocking did is it blocked out the variability that's associated with these two assistants. Okay, and that's the idea. We now can explain why, if on average all of these are lower than this one, it's okay. We can still look at the effect of the treatment. Okay, and that's the point of blocking. You come up with a factor that you think has a major impact and you block by it. And you'll notice that we repeated the design in each block. And so that's a characteristic that you'll see. And like I said, we're going to spend a whole lot more time on blocking. So if you don't have that completely understood at this point, I wouldn't expect it, and we'll cover that much more. Now, to look at that, again, if we're thinking about our experimental error, we have our treatment variability, which would be our water temperature. But now we've actually explained more of the variability in the block variability, which relates to the assistant. And if we hadn't done that, then that block variability would have been in that random unexplained variability, right? So our goal is to pull out as much information to explain as much as what's going on down here into this part. So again, this is showing you that if the block wasn't incorporated, then that block variability is just going to go right down here into the random variability. So a basic error control designs, the ones that we'll talk about are a completely randomized design, and that there's no blocking factor, and that's what we're going to start with is the CRD. We have randomized complete block designs, which are RCBDs. They incorporate one blocking factor. We're going to talk about Latin square designs that incorporate 2 factors. And we'll talk about a couple other designs as well that incorporate these kind of blocking ideas. So to keep kind of going with that, we want to talk about this idea of replication and randomization. So with replication, it's very important to look at replication, and it allows for the assessment of unexplained variability. So more replication means more precise inference. If you only have one replicate, it means that you can't do any inference. Okay, so you can't make decisions from your data. Now, I will say that some people have overcome that in certain ways, but just in general, replication is going to allow you to make your conclusions. Okay. The other important idea is randomization. So it provides objective approach to treatment assignment. It leads to statistical independence among your responses, and it averages out extra sources of variability. So I'd say that that's one of the major advantages of randomization. It's one of the main concepts of experimental design. Because it's just going to average out things that you don't even think about happening. So let's kind of go back. Let's just say we have the 6, right? Here's our 6 containers. I'm not going to have the blocking in here because we don't necessarily need it. So one of the first things I said is we were going to have the treatments, and we called it the water temperature, but I'm still going to go with A, B, and C, right? So this one receives the A treatment, this one is the B, this one's the B, C, and A, right? That was the randomization. So maybe it's that if you look in the room where this is actually being done, there's a window right here, right? And so there's sunlight that's coming in. Well, if I had just put A here and A here, B, B, C, C, just pass them out that way, then I'm not sure that maybe the reason that these are growing more is not the water temperature necessarily, because the light source here is actually changing the water temperature and affecting it. That's one thing I didn't think about before I started the study, but it could have an impact. If you randomize, you're hoping to average out some of those extra sources of variability. Okay, so that's the randomization. The replication here, and we'll see this on the next slide, but replication, if it's a balanced design. So balanced means that there's an equal number of treatments per EU. So here it's balanced, right? Because I have the treatment A is seen on 2 EUs, treatment B is seen on 2 EUs, and treatment C is seen on 2 EUs, right? So with replication, if it's balanced, you can always take your number of EUs divided by your number of treatments. So my number of EUs is 6 divided by my number of treatments, which is 3. So this tells me that I have 2 reps per treatment. That's what I see it here, right? Here's 2 reps of A, 2 reps of C, 2 reps of B. Okay? That replication, again, allows me to describe the unexplained variability. What I mean by that is that this container right here and this container right here should be exactly the same. We set it up, we put the water in, we start with the very similar homogeneous sets of shrimp. We put in treatment A, they both receive A. Any differences among these 2 EUs is background noise, unexplained variability. So the point of this is to say, okay, these should be the same. This is what I can't explain why they're different, but they're just different. Do I see enough of a change from this container to this container that's above and beyond the difference that I just see from the background noise, right? Is there a signal, the difference between A and C here, is that enough above what I'm just seeing from the background noise? That's what you're doing with these experimental designs. And that should actually clue you in that that's similar to what we talked about in Stat 5615. It's ANOVA, right? It's analysis of variance. You're looking at the signal compared to the noise. Okay, and so again, that idea of replication allows us to look at that. Okay. So here again, caution the subsampling. So if we have multiple observations per experimental unit, that doesn't necessarily mean that you have replication, and so people run into that problem. Replication means that you have to have duplicates of the treatment assignments to the EUs. So hopefully that example we just looked at is helpful for that. And then subsampling is duplicates of your OUs. So let's go back to the shrimp example. I'm going to go, again, forget about salinity. We're going to call it cold, warm, and hot. And for this one, we're going to measure 10 shrimp from each container. So there's 30 responses, okay? So just 3 containers, 10 shrimp from each container. So we have the 30. So our EU here would be the 3 containers of shrimp, and our OU would be the 30 individual shrimp. Here we have 10 OUs or subsamples per treatment, but we only have 1 replicate, right? So again, the idea here that we have the 10 shrimp here, 10, 10, people sometimes want to treat those as replicates. Those are not replicates. Those are actually just multiple observations per EU, right? And so here what you can say is if you take your number of OUs divided by your number of EUs, okay, then you get your subsamples. So our our OUs is 30 divided by 3, we have 10, okay? All right. So here's the shrimp example again. This is with replication. So we have 9 containers and we have 3 EUs per treatment, right? So here inference is possible. You'll notice that if you start counting your replicates, you see warm water 1, 2, 3 times, right? You see cold water 3 times, and you see hot water 3 times. So this is an equal number of replications per treatment. That's what we had just talked about. So that's, again, that's the idea that we have a balanced design, and we'll focus more on balanced designs. So now I want to talk a little bit about randomization. and how you actually do that. So on this next slide, it's showing you how you actually can randomize, and I will post another video. This can be done in Excel. It can be done in any software. There's random number generators even online, but I'll show you guys also in JMP software how to do this. This is kind of a complicated way of looking at it, and I think the example is more helpful, but we'll use this notation as we move throughout. So T is going to be our number of treatments, and N is going to be our number of replicates. So our total number of observations is going to be capital N, which is just our number of replicates times our number of treatments. So that's how many EUs we have. Right now, we're not going to worry about subsampling. So we're going to label our EUs from 1 to N. So if you think about the last one, we would label, these are our experimental units or the containers, and so we would label those from 1 to 9. And then we're going to randomly permute those numbers of 1 to 9. And for the first set of 3, they're going to receive the cold. The next set of 3 are going to receive the warm. And the next set of 3 are going to receive the hot. So if you look at it here again, we've done the EUs, and we randomly permute that order. So here's 5, 7, 8. So 5, 7, 8 gets cold. So if you look at it here, 5, 7, and 8 get the cold water. Now again, there's many ways that you can do this, but the point here is that this is a random allocation of the treatment combinations or treatment that we have here, which is the water temperature. And again, and looking at that, that's the important kind of aspect of it. Okay, and like I said, I can show you how to do that in JMP software as well. If you do it under blocking, it's important to understand that if we're going to use B as our number of blocks, each block has NT experimental units. So what you would do is you would just randomly apply that in each block. Okay, so here, if we were doing that, we would number from 1 to 3, 1 to 3, and 1 to 3. We now have 3 blocks, and we'd make sure that each treatment appears once or maybe multiple times depending on the design that you're looking at. And it turns out that you do actually have blocking here, or sorry, replication. So we can look across the blocks for our replicates. We still have 3 replicates of each of our treatments. So that kind of wraps up this intro lesson. So just to summarize, we talked about the idea of experiments versus observational studies. I tried to introduce some notation and terminology that we'll be using this semester. And then the last part of this was the focus of talking about experimental units versus observational units and some calculations and terminology that relate to that. So what we will do is I'm going to post a homework for you guys that will cover some of these ideas with the EUs and OUs and get more practice with that. And again, you have to step back and think. The EU is what the unit that I'm randomly applying the treatment to. So I always look for that. What am I randomly applying the treatment to? And the OU is the unit that you're actually taking the measurement from. It's not the measurement you're taking, it's the unit that you're taking the measurement from. And so that wraps up this kind of intro to experimental designs.